Unit 4, Population Genetics. This is when we'll be talking about some different concepts here with how populations work with our genetics. So for example, we're going to do some basic terminologies. First off, what's a population? Population is any group of organisms in a given time in a given area. So I could talk about all the people that lived in the United States in the 1820s and how that population was sort of seen based on height or socioeconomic class or any sort of data like that I want to look at. In this case, we're doing genetics, so I could talk about things like the amount of people have a certain hair color, a certain eye color, a certain weight ratio, different kinds of lengths of fingers, things like that. I could also talk about populations in an area like all the rabbits in Warrensburg, or all of the trees in the United States, all the oak trees in the United States, something like that. Those are all valid things that can be utilized. I can talk about a gene pool. Gene pool is all the genes within a population in a given time. So right now I can say all of the human population has a gene pool of all the different traits you can find in every possible human. Does that mean every person has all those traits? No, but these are all the options that are available. Population genetics then is, how do I look at population as it being affected by natural selection and evolution? So as the genes are moved around in population over time, that's going to cause some differences in the populations to the point that they can split and go towards one direction. Maybe all of the certain type of trait is eliminated out of the population, everybody becomes the same. That's going to affect that group of organisms. Or maybe it splits them into two groups. Group A and Group B have different traits. Over time, they stop recognizing each other and they become different species. So you can create natural selection and evolution that way. Allelic frequency or allozymic frequency. Allozymes are different versions of proteins, and you can get those through different protein foldings and things like that. So you can affect the way things work. We can analyze those ratios, or we can look at allelic frequencies. Alleles are different versions of genes in a population. So it could be two different genes in a population, or one gene in a population with two different versions. So you could have a nail claw that's either short or long, and those could be two versions, two alleles. Or it could be the traditional way of looking at like a pea plant, green peas versus yellow peas. Those are two different alleles. Same trait, pea color, different alleles. How do I calculate that though? That's going to be the question we're going to have to figure out. I also need to look at how the locus is. The locus is the location on a chromosome that these alleles and these genes are located. So it could be on the third arm of chromosome number 5 or chromosome number 6, or it could be on multiple different types of genes. Therefore, in that case, we would say it's loci because it's in different locations, because the plural would be loci. So how do I calculate the allelic frequency? Formula is very simple. P plus Q equals 1. It is something many people who have taken a trigonometry class are going to see because it's a binomial probability theorem. So if you know P, then you know Q, because if P is 0.4, then Q is just 1 minus 0.4. 4.4 from 1 is 0.6. So it makes it simple. You can go back and forth either way. As long as you know those numbers, it's pretty straightforward. But how do you get to those from a population without actually looking at everybody's genetics 100% all the time? Well, you can do it through looking at what we call genotype frequencies. Genotypes are what the genetics show, using traditional Mendelian concepts like dominance and recessiveness. So you have homozygous dominance, big A, big A. Homozygous recessive, little a, heterozygous, big A, little a. I can use that to calculate this if I know those values. So for example, population has big A and little a as alleles, then I can use this concept to figure it out. Here's how I do it. I make an estimation of P. That's why I have the brackets on here, because that means it's not the actual number, it's an estimation of the population based on the information I'm given. So my estimation would work like this. 2 times the homozygous dominant, big A, plus 1 of the heterozygous. Makes sense because there's 2 of the big letters and only 1 of the big letters here. Kind of add up the big letters here for given P. I chose big letters P. You could do it the other way around, but typically this is what we do, use the dominant letter as the letter P. Divided by 2 times the total number of individuals. That'll give me my value. So if I know this P, then I can figure Q out. I can do the estimate of those. I can also use to calculate other things, like variance. Variance can be calculated by doing this. The variance of P is going to be my estimate of P times the estimate of Q, which is 1 minus P, divided by 2n. That'll give me my value for the variance. If I know the variance, I can calculate things like standard deviation and all the statistical tests that you can do, like correlation and so forth and so on, t-tests, everything else. So let's do a practice problem here. Show how this works. Here's my values. Homozygous dominant, 250 individuals. Heterozygous, 150. Homozygous recessive, 200. Total number, 600. I have all the values I need to solve the problem, to figure out what A is, what little a is, what the variance is, and then how can I do other measures off of this. So let's do the first one here. Frequency of A. We'll assume this is P. We'll assume this is Q. I'll leave my brackets off for now. So to figure out P here, I'm going to need 2 times the big A. So it's going to be 2 times 250 plus 150, all divided by 2 times 600. So I'll just put 1200 here because that's the number for it. So I can solve this out. That would be 500 plus 150. So it's going to be 650 over 1200. Well, those numbers I can figure out pretty straightforward. I can use a calculator to figure it out. 1, 2. I know it just passed 50%, obviously, because 600 out of 1200 would be 50%. So it's just slightly past it. In this case, it happens to be 0.54. Well, if P is 0.54, then Q is easy enough to figure out because it's just 1 minus 0.54, which is going to be 0.46. I take these two numbers. I can figure variance off that by taking this times this, and then divide it by 1200. It's going to be a small number of 0.0002. Very small variance I can then use to calculate things like standard deviation and everything else. Other measures then I can calculate, yeah, easy. I can just take the square root of that to figure out my standard deviation and so forth and so on to be able to do all those other variables. Another question happens is, what if I have more than 2 alleles? Because there's some things we know have more than 2 alleles. Well, it just follows the same probability theorem. Everything's still of 100%. So, if I have multiple alleles, for 3 examples go like this, I add another one. P plus Q plus R is equal to 1. In this case, though, I need to know 2 of the other allele frequencies in order to calculate the third one. Because I know 2, I can always calculate the third one, simple enough math. So, like for example, if I want to find out R is just 1 minus P plus Q combined. So if I knew this was 0.3 and this was 0.4, that's 0.7, that means R is going to be 0.3. It's just what's left over. And you can do this with 5, 6, 7 alleles. It becomes very complicated, but you can do with all of those things. And that is the end of our notes today.